Let me welcome everybody to today's webinar. This is brought to you by the Tennessee Urban Forestry Council and the University of Tennessee's Residential and Community Forestry Working Group. My name is Neil Letson. I serve on the board of the Tennessee Urban Forestry Council. I'll be your host this morning or this afternoon, depending on what part of the state you're in. Where today's topic is going to be on urban tree pest, what to expect in 2022. And that would be for Tennessee. And I'm really looking forward to this presentation because we don't get very often a look into the future, at least a heads up on what to expect, but that's what we're going to do today on pests that might be a problem with your urban tree and how we can respond and things that we can do to hopefully mitigate that problem. So this is a real timely topic for this time of year. Now, for those of you joining us for the first time, I do want to mention two things, kind of housekeeping. Today's webinar does qualify for one ISA Certified Arborist CEU. So if you are a certified arborist in need of CEUs, this qualifies. But you have to do a couple of things beforehand to get credit for that. Number one, when you registered for this webinar, we needed to know your name and also your certification number. If you did that, you don't have to do another thing. We'll fill out the forms and send it in for you. But we will check to make sure you sit through the presentation. So make sure you sit through and pay attention throughout this next hour. If you are a certified arborist and still need a CEU but did not give us your complete name or your CEU number, I think Alyssa has posted that on the instructions on that here. And let me look at those. You need to put your name and the CEU on there. Step one, click on the participant's icon at the bottom of the window and then find your name that you submitted, and if you need to put in a full name, you can rename that and then type in your name. And Alyssa, can they type in their certification number during this process?
Yes, they can.
Okay.
And here
great.
I'm going to put my email address on the screen. If you need, if you need to send me your number separately or just double check that we got your information.
Okay. I know you're going to get great value out of the presentation regardless, but do want to make sure you get credit for your certification CEU. And then the last thing, we will have a short period at the end of the presentation for questions and answers. And the way we handle that is throughout the presentation. We're not going to ask you to announce or read your question. Just enter the chat room and write your question in. And at the end of the presentation, Alyssa will read those off and our presenter will provide an answer. So we want to make sure that anything not covered or anything that you want more details on or didn't understand, you will have an opportunity to do that by using our chat room. So with that, I'm going to introduce our speaker, Dr. Ellie Lopez, and she's a very personable person. She said to call her Ellie or Eleanor. So I appreciate that. And she's going to be our speaker today. Eleanor is a recent employee of the University of Tennessee's Soil, Plant, and Pest Center at Ellington facility here in Nashville, just north of Murfreesboro, where I am. And her job involves diagnosing plant pathogens, identifying insect pests, and assisting with soil analysis, all to try to transfer her knowledge and expertise in a way that helps the clients, the homeowners, the practitioners and professionals that deal with these problems, but need help in a proper, accurate diagnosis. And that is her job. So Eleanor, that's all I'm going to say about you, but I would like you to fill in some of the blanks and help us understand a little bit more and know you better.
Awesome. Thank you, Neil, for that introduction. And thank y'all for having me. I'm going to try and share my screen while I believe y'all still have it shared. Let's see if it works. Okay, there we go. Everybody see it? Everybody can see my screen? Yes? Okay, awesome. So, yes, I'm Eleanor Lopez, and I am the Plant Pathogen and Insect Identification Diagnostician for the University of Tennessee Extension. You can find my lab on the Ellington Ag campus here in Nashville. So I'm excited to talk about my lab some and some urban tree pests that we can be on the lookout for in Tennessee in 2022. So I don't know why I'm seeing, should I admit this person? Can you see this person? Okay, thank you.
Sorry, you can ignore this request.
All right.
I'll do that on the back.
All right, thanks, Alyssa. So I'm from Nashville, so it's really nice for me to be back home in Tennessee. I did my undergraduate work at the University of San Diego, where I majored in Spanish and environmental studies. I also got to swim Division I out in San Diego. Butterfly was my main stroke, so I think it was a little bit of foreshadowing for me becoming an entomologist. It was great. You know, we love the beach, we love the ocean. I learned a lot about climate change and other sorts of things out there that really drove me to want to get more involved with food systems. As a swimmer, I can eat a lot. And I think that's one thing we can all get behind, is having clean, healthy, accessible food. So I moved back home to Nashville and I went to Lipscomb University, where I did a master's in sustainable food systems. And I learned a lot there. I got to meet a lot of wonderful people working toward supporting local food systems in Nashville, but that program was very systems based. So looking at not just growing food, but food waste, energy that goes into food systems, packaging, transit, all sorts of things. So it was really nice to get a big picture about how systems work, but I realized that what I was really most interested in was the plant part of the food system. Protecting our plants, how to make sure we're growing healthy plants, and knowing what's wrong with our plants. So that led me to the University of Florida, where I was attracted to the Doctor of Plant Medicine program. So this is more of a professional doctorate based on heavily on diagnostics. So diagnosing plant pathogens, our nematode pests, our insect pests, and also learning about plant physiology and soil science that are all factors in whether or not our plants are able to absorb nutrients and stay as healthy as possible to be more resistant to pest pressure. So that was my doctoral degree. And while I was doing my doctoral degree, I was also doing a research master's in entomology and nematology, looking at a novel tree crop in Florida, olives. Most of the time we think of Florida, we think of citrus, but there is a very serious insect pest of citrus called the Asian citrusillid that transmits a very serious pathogen of citrus greening. So a lot of citrus growers were abandoning their fields, not able to manage the pest pressure and the disease pressure from this insect and pathogen. So I was working with some growers on olives to see if they could replace some of the citrus groves. So I love working with trees and pests of trees, and I learned a lot there. And it led me to this job here, which is great because I get to use all of my skills that I learned. I get to do plant pathology, insect ID, a little bit of nematology, and help out in the soil lab when they need me. So again, our lab is located on the Ellington Ag Center at 5201 Marchant Drive. So y'all can email, we accept samples that are emailed. Mailed physically to the Plant and Pest Center. You can also drop them off. And we also have portals for our local county agents and our Tennessee Department of Agriculture plant inspectors to upload images for diagnosis online. So sometimes things can be diagnosed just by an image. A picture of an insect, I can more often than not identify from just an image. But when we get into our plant pathogen samples, that's when I need a physical sample mailed in. So I'd like to encourage everyone on this call to contact your local UT TSU extension agent in whatever county you're in, because they are able to, if you just have a picture of an insect, of a diseased plant, of a plant problem, upload that to me in the lab and I can then review it. And this is a free service through the extension office. So our other services are for a fee. Plant pathology diagnosis is $15, and insect identification is also a $15 fee. So it's the same fee as paying for your soil to be analyzed to identify what plant problem or insect pest you may have. We also offer a few additional services for our golf course managers. And again, we have our free distance diagnostics service through your county agent office. So when thinking about distance diagnostics, there's a great resource at this website to find your local county agent if you'd like to submit a picture for analysis. Good distance diagnostic sample are distance diagnostics is best suited for insects. So when I'm looking at an insect image for analysis, I need to see certain features like antenna, the wings, mouth parts. All of this is very important. And this is also why it's great to bring a sample or to show your images to your extension agent, because a lot of our agents are able to take images more close up of our insects and other problems. A lot of our agents have microscope cameras and higher magnification cameras available for their use. It's really great to always take pictures, whether it's an insect or a plant disease, with something to scale, so I can get an idea of just how big is this insect or how big is the leaf spot. So having something of a known size like a quarter or even a ruler in an image is always going to be helpful. Other important information that you can submit when uploading an image for diagnostics are things like, what is the host plant? Sometimes just a picture really close up of bark, I'm not going to be able to identify what plant I'm looking at. So taking a picture of the entire plant, of the entire growing area, gives me a lot more information about other environmental factors that could be causing stress, plant competition, too much shade, to being close to something like a downspout on a building causing too much water sitting in the soil. All of those bits of information are really helpful if I get more images of the entire growing area. And then particularly with insects, having some images of the damage that's been done by the insect is very helpful. So checking the entire trunk of a tree for any boring holes in the tree will let me know, which type of wood boring insect we may have. Is there only damage to the leaves? What does the damage look like on the leaves? Are there shot holes, or is it more what we call window painting, very finely, where just the top layer of the leaf is eaten? So all of those other bits of information can be really helpful for me when trying to identify what insect pest we may have from a picture alone. What's not so good with the distance diagnostics is plant pathogens. Plant pathogens, I usually do need a physical sample. And what's really important when I get a lot of, my tree died overnight. What's wrong with my tree? And a lot of the times when that happens, when our tree just completely wilts and the leaves are still on the tree, is that there's a large wound on the main stem, or there is root rot that is preventing the flow of nutrients and water up to the canopy. So when we're looking at a plant and it looks sick, we always want to check for major wounds to the main stem and large lateral branches. And we also want to check the root health. So if our roots are white and healthy looking, that's great. If they're brown, if they're easily pulling off when we excavate and take a look at some of the roots on our larger woody plants, that's when we're going to want to send root tissue in for analysis. And sometimes plant diseases can actually be insect damage. So we always want to check for insects. So especially always checking the bottoms of leaves. A lot of the times our bugs like to hang out on the underside of the leaf. And we also want to assess the environmental conditions. So this goes back again to where images of the entire landscape can be helpful. Sometimes things like rock mulch can overheat in the summer and burn the roots of our larger plants. And sometimes they can even change the pH of plants. A rock mulch like this might increase the soil pH for an acid-loving tree or shrub, which will make it very difficult for that plant to absorb the nutrients it needs. So I'll definitely recommend at least having a little pocket magnifying glass when we're going out to look at our plants to check for insects. And this is just another example of why it's very difficult for me to identify the causal agent of a plant pathogen from an image alone. So these are two different fungal pathogens on hydrangea. And one thing that I think is extremely important is making sure that we identify what the real pathogen is here so that I can give the most effective science-based recommendations on managing the pathogen moving forward. There's a difference in efficacy for different types of pathogens. So another pathogen that can cause these sort of leaf spots on hydrangea are bacterial pathogens. So what works for a fungal organism is not necessarily going to be effective for a bacterial organism. So in a case like this, I always want to have a sample in the lab so that I'm able to give effective management recommendations. And I wanted to talk a little bit about physical sample submission. If you find an insect pest on a tree, you are welcome to submit it physically to the lab. We do also at your local county agent office have vials of propylene glycol, which is a very safe material to send in the mail. So this is a great way to send adult insects, immature insects like caterpillars, grubs, other larvae. I also accept sticky cards. Hanging yellow sticky cards is a great way to monitor for insect pests. So if you ever are monitoring for a particular pest of concern, whether it's borers or moths, sticky cards or what you've collected from a trap can be sent to the lab for identification. And we always want you to include a submission form when submitting to the lab as well, so we can get all of that good background information. Where on the plant was the insect found? What was the damage being caused? When was it noticed? All of that is very helpful for me to know. For plant samples submitted to the lab, I really need plants that have some healthy tissue and some symptomatic tissue on them in order to make a good diagnosis. Unfortunately, I am not a plant mortician and I can't give an accurate diagnosis if the plant is already completely dead. There are certain pathogens that will be present on dead tissue alone, so they're feeding on the dead tissue and they can outcompete, once the plant is dead, a pathogen or problem that originally caused the plant to die. So we always want some healthy growth and some symptomatic tissue on plants submitted. It is always helpful to have a handful of fine feeder roots to rule out root rot pathogens. So even on our large trees, just excavating around the base of the tree, pulling some fine feeder roots, that's going to be really ideal if we're seeing serious decline in the plant health. When submitting samples, if sending in the mail, for woody ornamentals like trees, taking clips of the branch tissue and putting them in a plastic bag is great. If you're able to send a whole seedling or sapling to the lab, putting the roots in aluminum foil or keeping them separate so the dirt isn't getting up onto the plant is always ideal. If you're working with someone who's growing fruit in an urban setting, so if we've got someone growing a small urban fruit garden, and the fruit is showing signs of disease or pest pressure, we want to wrap those in newspaper so that they don't rot in the plastic. And these are just some more notes about our submitting a sample. It's very important for me to know everything that's been done to the trees before they get to the lab. So because some of these practices, some of the historical information about chemicals used, about irrigation, can help me give better management recommendations in terms of how to reduce pest pressure. Sometimes over-fertilization can attract more insects to a plant. Sometimes overhead irrigation can cause an increased amount of fungal and bacterial pathogen spread. So the more I know, the better recommendations we can give. Sometimes I will find no pests on plants submitted. And oftentimes this can be because there is a problem just in general with the soil. So sometimes I'll recommend a soil test be done if I suspect it's a nutrient problem and not a plant pathogen or insect pest problem. And sometimes our plants are more susceptible to environmental stress. So this does happen occasionally. So now I'll get into some of the tree pests that I see frequently in my lab and that you can anticipate in the coming year. So one very frequent flyer in my lab are pin oaks. So pin oaks are a member of the red oak family, and they are particularly susceptible to a bacterial pathogen called bacterial leaf scorch. This pathogen we don't currently offer serological tests for, and it's something that I'd really like to start offering by the fall. The fall is when you'll see symptoms of leaf scorch on the pin oaks. So you'll see very brown leaf margins where it looks like there's, it gives it that scorched appearance. Oftentimes we'll also see leaf spots associated with the leaf scorch. Tubacia leaf spot is another very common pathogen in the fall on red oaks. It's not a significant pathogen. The leaf spots are not something that I'm worried about, but the bacterial leaf scorch eventually will kill the tree. So keeping the trees as healthy as possible, depending on their age, is important when we suspect we have bacterial leaf scorch in the landscape. This pathogen is also seen on other oaks in the red oak family, and I have seen it pretty frequently on sycamore trees as well. So this is a big one, and you'll start to see dieback symptoms and leaf scorch symptoms in the top of the canopy, and it will spread down. Laurel wilt is another very serious pathogen of sassafras and spicebush. This pathogen is spreading throughout our state. It is spread by the red bay ambrosia beetle. So you will see frass tubes and signs of beetles entering the trees. And when we scrape, and you'll also notice this on sassafras especially easily, as the trees will appear dead and the leaves will still be holding on to the trees though. So they'll be kind of dead, brownish red leaves still clinging to the tree. And this is a good sign that there may be a laurel wilt infection. So when we scrape down the bark on the main stems of sassafras spicebush, we'll see some pretty distinct discoloration in the sapwood. And this is an indication that we've got laurel wilt. So this one's a pretty big concern, more in our forested areas where we have a lot of sassafras. But if you're planting some of our native plants like sassafras in urban landscapes, this is something that we'd like to be on the lookout for. Evergreens. I get lots and lots of evergreens in the lab, lots of arborvitae, lots of juniper, and there's a couple of key problems that are pretty consistently found on these evergreens. Root rot is a big one. When you go and you're selecting your plants, always check the roots for root rot. This is a very common problem, especially in big box stores. So always, 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 before you buy or plant your plants, check for root rot. These roots are brown, rotting. We want them to be white and healthy. So this is a big one. Phytophthora root rot is one of the biggest problems I see on almost all of our evergreens. Also see a lot of spruce spider mite and two-spotted spider mite feeding damage on our evergreens. And these mites can be present all year round, and they lay lots of eggs on the plants. So making sure that we are monitoring for them is very important all year round. Water stress is a big source of decline in evergreens. A lot of people like to plant nice, big, beautiful Leyland cypress and thuja species as screens. And eventually these trees will grow to be quite large, and they end up competing with each other for water and other nutrients. And this stresses the plants out. So making sure that we're planting them far enough apart from each other that they're not competing for light, for water is important. And water stress can also be too much water. So too much water in the landscape can cause these plants to be more susceptible to root rotting pathogens. Fungal cankers are very common on evergreens. Phytophthora root rot can cause cankers farther up the tree. I see a lot of Ceridium canker, a lot of Botryosphaeria canker, and the cankers are more associated, they're usually associated with stressful planting conditions as well. So we always want to be examining branches and the main stems for any sap oozing, any bleeding. Those are good signs that we have a canker pathogen. And pruning out any discolored tissue, any tissue that's completely browning out is really important to reduce the spread of fungal cankers and to keep our plants healthy. So when we're pruning, we always want to make sure that we're pruning well below, a couple of inches below any discolored tissue, and cleaning our pruning shears between cuts so we're not spreading these fungal cankers to our other plants in the landscape. Tip blights are also some things that are more infrequently seen in the lab. And some of these evergreens can be very susceptible for herbicide injury. We get questions fairly frequently about herbicide injury in the lab, and unfortunately, I cannot test for herbicide injury, but we do get plants that are definitely symptomatic of herbicide injury. And the Tennessee Department of Agriculture can test for herbicides if this is a problem that's suspected. I get a lot of Japanese maples in the lab throughout the year. A lot of the times they'll come in with this very bleached white leaf tissue. And these plants are very sensitive as well to environmental extremes. So right now is a really good time to be thinking about any young trees that we're planting. We want to protect them if we get unseasonal cold snaps, because that will cause burning symptoms like this on some of the new foliage that the trees are putting out in the springtime, and also affect more strongly our more sensitive plants. There are some other pathogens that can cause these sort of scorched samples on Japanese maple. So it's always good to submit a sample to the lab if you're not sure. But our Japanese maples are very sensitive. A couple of other things to keep in mind when we're taking care of our plants are mechanical injuries and injuries caused by non-insect or disease pests. So this was from a black walnut grove, and one of the big problems here is there's no mulch around these trees. So what happened here was this person was mowing around their trees and their lawnmower hit this tree, and this giant canker is what resulted from being injured. So making sure we've got the right amount of mulch around our trees to not only suppress the weeds, but reduce this type of injury is really important. Because when our trees are impacted by this kind of mechanical injury, they become more stressed, and certain insect pests and pathogens are more likely to infect our plants when they're stressed, because the plant is producing chemicals that are related to the stress that attract pests more. So always making sure we're mulching correctly and not accidentally hitting our plants with our tools, injuring our plants with our tools is important. Another really common problem I see on a lot of different plants is woodpecker damage. So this is really characteristic of the yellow-bellied sapsucker woodpecker. And these birds are usually getting into our pines, some of our other plants to feed on the sap of the trees, or they're feeding on insects that are in the trees. So they can be a sign of an insect, like a wood-boring pest infestation in our trees as well. So to protect our trees from this sort of damage, our trees will heal from these sort of mechanical wounds on their own. But one thing we can do to prevent things like sapsuckers from coming back is to cover these wounds when we know that they're going to be in our state. So from about now until June is a good time to cover these holes in our trees to prevent further feeding. Another really common disease is cedar apple rust. We will see these sorts of tumorous galls on cedars starting in the early spring once we get some nice wet weather and warmer temperatures. The best thing to do when we see these galls on our juniper, on our cedars, is to prune them off, because once these start producing spores, they will spread to our rosaceous hosts, which this pathogen has a really interesting relationship with. So our apples, our hawthorns, crab apples, they will become infected by what this big gall will release. And you'll see bright orange leaf spots on the rosaceous hosts once we've seen these galls release their spores. So there are some protective fungicides available for cedar apple rust on the rosaceous hosts. So if you're dealing with any of these hosts, and we've got any cedars or junipers nearby, this is definitely one to watch out for and to plan for. And we've got some great extension publications with spray schedules and other sorts of good recommendations for this particular pathogen. Mites are another big pest that we see all year round, and mites are going to be on basically every single plant that we grow in our urban landscapes. Two-spotted spider mites and spruce mites are the most common on evergreens, ornamentals, fruits and vegetables. And they're super cute and super tiny, but they can feed a lot. A very good indication on our broadleaf plants of spider mite feeding damage is this very stippled look. So it looks like someone's kind of just taken a pin and prick, prick, prick to the top of the leaf. Horticultural oils, insecticidal soaps are really nice, not harsh chemical alternatives to use on these pests. It's really important to know the temperature when we've got a mite infestation. Oil sprays are really great at suppressing mite populations, but we want to make sure that we're using a dormant oil at this time of the year. So when we're still in our cool temperatures, as opposed to a horticultural oil. And that's because we don't want the oils to burn the plant or injure the plant. So making sure we're always reading our labels when we're dealing with mites is very important. And our symptoms look a little bit different on evergreens. So mites and many insects like to feed on that fresh, succulent, young foliage that's being produced. So we'll often see tip dieback on evergreens from mites feeding at the very tips of the branches. A great way to test to see if you've got mites on your tree is to take a white sheet of paper and just tap the foliage onto the white sheet of paper and then circle the tiny little specks that fall off. If it's dirt, it's not going to move. But if it's mites, they'll move outside of the circles and you'll know, okay, I should probably look at some options for managing mite populations so we don't have this dieback on our plants. Aphids are another big insect pest that I've seen in the lab on just about everything. Aphids have these cute little tailpipes at the end of them. So that's how we identify aphids as opposed to other insects. These insects have little piercing sucking mouth parts that will suck the chlorophyll and sugars out of our plants and can sometimes cause browning, and really the saliva equivalent that insects have can be very irritating to plants and cause puckering and malformation of the plant tissue. So in our high value plants, we sometimes want to use a systemic insecticide to reduce that intense plant reaction. But there are horticultural oils and soaps that will also reduce aphid pests on our larger plants. And this is where removing weeds is really important for our foliar feeding and pests like aphids. They like to hide out on a lot of them feed on a lot of different host plants. So making sure we're keeping our weed pressure low is important in reducing pest pressure here. Another important pest that is in our state now is the crepe myrtle bark scale. These scales can completely coat our crepe myrtles, and similar to aphids, they have piercing sucking mouth parts. So they just kind of hunker down once they become adults and suck all the plant sugars out as quick as they can. And they produce a lot of sticky excretion called honeydew that then fungus will grow on. So when we have these really heavy foliar and bark insect pests, that honeydew covered by fungus can sometimes coat the leaves, and that can eventually lead to plant death if the leaves are covered and no photosynthesis is happening. These are very white, fuzzy insects, and we can know that they're crepe myrtle bark scales because they're very pink when we flip them over, and they lay these beautiful pink eggs. And starting in early March is when we want to treat for these, and there are some more systemic insecticides available for these as well. Another important insect pest that is, I suspect, is going to be pretty widespread throughout the state soon is the emerald ash borer. I'm happy to say I haven't had too many ash samples with emerald ash borer damage in my lab yet, so that makes me optimistic that our wonderful tree care professionals are protecting our ash trees and doing a good job of that. But this is one to definitely anticipate. A good sign that we have emerald ash borer is this sort of D-shaped, so it's not a perfect circle, it's kind of a D-shaped exit holes from this pest on the main trunks. Oftentimes this pest can be tricky to catch because they like to feed at the very top of the crown, and then the damage spreads down. So if you've got ash trees, making sure that you're protecting them at this time with a pest management professional is very important. And this is another important insect that is not here, but we definitely want to be on the lookout for: the spotted lanternfly. Although a very beautiful bug, This is a very serious and threatening pest for our state because it has an incredibly wide range of host plants. The primary host of this insect is the tree of heaven, which is considered more of a nuisance plant, but it can feed on a bunch of different plants. So we really want to protect not only our landscaping and our tree plants, but our food plants from this pest as well, since it can feed on so many different things. So if you think you see this pest, please let me know. This is definitely one we want to be on the lookout for. And just a note on our pollinators, we always want to be pollinator friendly when we're applying any sort of systemic insecticide. I know a lot of our high value plants, our beautiful, rare, ornamental plants, we sometimes need to protect from things like emerald ash borer with systemic insecticides. And we always want to make sure that we're applying those when the plant is not in bloom, so that our pollinators are not taking up that insecticide, and so that it's only targeting the insects, the serious insect pests that we want it to target to protect our plants without harming our wonderful pollinators. Just in general, in my lab, I'm really considering having our distance diagnostics portal be available for our landscaper clients from some of our more commercial clients to use. So that's something that I'd appreciate feedback on, but it is a free service at this moment for our extension agents. And I'm very sad to say that our entomologist in Nashville and our plant pathologist are retiring this year. So I'm excited to begin the search to find their replacements, and groups like y'all who our specialists really interact with more and need to provide educational materials for. I'd really appreciate any feedback y'all have about what you would like to see more from us at the Plant Pest Lab, and from our specialists who work especially in Tennessee and help with ornamental insect and plant pathogen pests for all of Tennessee. So thank you again for letting me come and speak with you. And you can always email me or find me on Twitter. So thank you again. And if you have any questions for me, it looks like there was one in the chat.
Yeah, we got several in the chat actually. Sorry if I can get my mouse to work. So you just were talking about pollinators. This one question, can you test for a honeybee herbicidal death or injury?
No, I cannot.
Okay.
I'm not even sure if anyone does that. That would be something I'd have to look up if that's even an option. I've seen some papers on it, so I know people are researching it for sure, but.
Okay.
Mm-hmm.
Another question. Some tree insects are beneficial to the environment. How do we respond to problem pests without harming the good ones?
Yes, it depends on the pest. So what's nice about some insecticides is they're very targeted for their pest. So if we had a caterpillar, let's say, that was defoliating a tree, we could spray Bacillus thuringiensis, and it will only affect caterpillar pests and not our pollinators, because it's very specific in the way that it works. It will only affect the particular cells within the gut of the caterpillar and not something like a bee or a praying mantis. So this is an area that's expanding with using more biological controls, with using more very specific technology to only affect the pest insects. That I think is a really exciting part of scientific exploration that will hopefully be more common soon.
Okay, thank you.
Mm-hmm.
Another question. Sometimes I see fungal bodies on the outside of tree surfaces. Can I submit an image to your lab for ID?
Yes. So those larger, more macro fungi are usually a sign that the tree is in decline. Some of them are not life-threatening for trees, but we're happy to identify them if possible. Sometimes we do need the mushroom sent in, but yes, if you want to send them in through your local extension office, that's the way that we like to receive them. But yes, you can always send them in.
Okay, great. Thank you. What do you mean by serological testing?
Yes. So some pathogens, I'm sure y'all have heard of the, so what I mean by serological is protein based. So when there are some tests that we do for certain pathogens that we can't grow outside of the host plant. So there are some fungal pathogens, there are some bacterial pathogens that we can isolate on a petri dish and to confirm that they're within the plant and they're the agent causing the disease. Some pathogens we cannot grow outside of the host plant. So we need to use a test to detect the specific proteins associated with that pathogen in a quick way. So we use tests that are very similar to like a birth control test or a rapid COVID test, where we use a buffer and we macerate some of the plant tissue, and then we're able to quickly use a color change to see, is that protein that's very characteristic to this particular pathogen present in this plant tissue?
Gotcha. Okay. Another question, do you have any recommendations on slowing the progression of bacterial leaf scorch in affected pin oak?
Unfortunately, there's not many, certain companies will offer things to try and prolong the life of the trees. I've not seen many studies showing the efficacy of these sorts of treatments. So really, the only thing to do is just give the tree all the TLC that you can. So sometimes I'll recommend a soil test to see if there's any nutrient deficiencies that could also be at play that could be corrected to try and make sure the tree is as vigorous as possible. But the pathogen, unfortunately, is spread by an insect that's a very strong flyer, and that's not really possible to manage. So it is, sorry?
Yeah, just making it as healthy as possible, like you mentioned. Okay, I've got a couple comments about how helpful the SPP Facebook page is. Your office's service in conducting identifications on pictures submitted through the Facebook site has been very useful. Are you continuing this service?
Yes. So this is where it's going to be tricky for me with my specialist retiring, because they really helped me out with that Facebook service. And it's something that I would like to continue. But it may be that we direct people more to their county agents to send those in, because we'd really like for the county agents to also be aware of what's going on and not just us. But yes, the Facebook page, I'm glad that it's useful. And we always want it to be useful.
All right, that is I think the last of the questions in the chat, unless anyone has some final questions or remarks. I can hang on here for a minute.
Okay, well, I think that ends our Q&A session. Ellie, that was a fabulous presentation and very informative. And I want to tell you and assure you that the audience here is a small representation of the interest people have in their urban landscapes, and trees are a big part of it. It's growing leaps and bounds. Middle Tennessee is your demand is going to increase, and I'm a little bit concerned that you're losing some staff people, and a lot of this is going to fall on your shoulders. So we need to do whatever we can to voice our support for this service y'all provide. And maybe that's something that some of the folks here that are watching can pass through the extension unit to let folks know that this is an important service. And I personally have benefited from it. But it's a great presentation. Y'all do a super job. And we just need to get you as much help as we can as soon as we can. So with that, we're going to close. I'm going to leave Eleanor on the line. There may be some lingering questions that come in, but Alyssa, maybe we could talk about next month. Did you prepare a slide for that, or do I just need to?
I did not. Yeah,
Okay.
apologies. I did not do that.
No, we're still fine tuning this operation, but our next webinar is March 17th. And the topic is going to be Tennessee's Champion Tree Program, where we aggressively look for the largest tree of each species. And we have a nice program that is administered through UT School of Forestry and Wildlife and Fisheries Sciences Department. And the young woman that runs that program is going to be our speaker. Her name is Jacqueline Payne. So if you're interested in the biggest and best trees in Tennessee, you'll definitely want to sit in on this presentation, and maybe you can help us expand our list. So we have other interesting topics coming up. We have a few open slots later on this year. If you have a suggestion for a topic or a speaker, please let us know, and we'll do our best to provide that. So once again, Ellie, job well done, and you have our support for doing what you're doing. Just let us know how we can help. So with that, any other questions, Alyssa? Is that it? Okay. So everybody that's still on that needs your CEU, we will take care of that and submit it to ISA. And thanks again for watching, and thank you again, Ellie.
Thanks, y'all.